All right. Good morning, everyone. Thanks for joining us, those of you who are here, and for those of you on Zoom. Welcome to a new academic year of MCC seminars. We're excited. Hopefully, we will get more and more people in person. My own personal plug for people getting butts and seats and finishing all the treats. So this morning, we're fortunate to have Dr. Susan Arnold with us. Dr. Arnold is an associate professor of veterinary neurology and neurosurgery in the Department of Veterinary Clinical Sciences at the University of Minnesota, and she is the new director of the University of Minnesota Canine Brain Tumor Program. The program investigates novel translational therapies for glioma by providing these novel therapies as treatment for pet dogs with high-grade glioma. Also, to optimize the canine model of glioma, her research focuses on breed-specific characterization of canine glioma. Today, she'll be providing an overview of the canine high-grade glioma model.
Please use the mic.
Oh,
Thank you.
sorry. Can you hear me now? All right, let's try this again. So today we're fortunate to have Dr. Susan Arnold presenting. Dr. Arnold is an associate professor of veterinary neurology and neurosurgery in the Department of Veterinary Clinical Sciences. She's also the director of the University of Minnesota Canine Brain Tumor Program. The program investigates novel translational therapies for glioma by providing these novel therapies as treatment for pet dogs with high-grade gliomas. To optimize the canine model of glioma, Dr. Arnold's research focuses on breed-specific characterization of canine glioma. Today, she'll be providing an overview of canine high-grade glioma model, the University of Minnesota Canine Brain Tumor Program, and the first breed-specific characterization of canine high-grade gliomas. Please join me in welcoming Dr. Arnold.
Thank you for the introduction. Can you hear me in the back? Great. And thank you all for being here today. And thank you for those of you who are tuning in on Zoom as well. I am perpetually very excited to talk about my research. And today I'm going to be talking to you about a number of things, but primarily focusing on breed-specific canine high-grade glioma and systemic immune transcriptomes. And I'm noticing that our screen went out. There we go. Okay, great. We're back. So I have divided this presentation into 3 different sections. Part 1 will be a section focusing on the background and significance, hopefully to get buy-in regarding how important the canine model of high-grade glioma, specifically glioblastoma, is. And then I will delve into the summary thus far of canine glioma transcriptomes, specifically looking at French bulldog high-grade glioma transcriptomes. And then I'll finalize by going into some of the preliminary results that we have for the canine peripheral immune system in the presence of glioma. I'll summarize everything and then tell you where I'm looking to take this research going forward. Now, I'm a veterinarian, and so I live dogs, cats. It's my whole life, my whole professional life, I should say. But I recognize that most people in this room are not veterinarians and don't think about dogs and cats the same way that I do. And I thought that it would just be a fun thing to throw in a couple of fun dog facts at the start of every new section. So we're going to go into the background and significance of this research. To get us started, my fun dog fact number 1 for you is that a greyhound can run 35 miles per hour for 7 miles straight. For those of you who are runners, that is a 1 minute 45 mile pace for 7 miles. It's amazing, just amazing. They can outrun a cheetah. But what we're actually here for is to talk about gliomas. Gliomas are primary brain tumors that arise mainly from neoplastic populations of astrocytes and oligodendrocytes, creating tumors called astrocytomas or oligodendrogliomas, respectively. They can be further divided into low-grade and high-grade tumors. Grade 4 glioma is called glioblastoma. I'm going to abbreviate glioblastoma going forward as GBM throughout my presentation. And glioblastoma is the most common primary brain tumor in people, and it's also the deadliest brain tumor in people. Even with the current standard of care, which consists of surgical resection, radiation therapy, and temozolomide, the median survival time is 14.6 months, and the 5-year survival rate is less than 5%. In the past 30 years, we have seen 7 United States presidents the development of the internet. Pluto, pictured here, is no longer a planet. We have self-driving cars. The Human Genome Project has been completed. Overall, we've seen amazing advancements in the past 30 years. Yet in those same 30 years, despite extensive efforts, we have seen no change in the glioblastoma patient survival time. These are 5 men who all died from gliomas. I'm imagining that you recognize some of the people pictured here. Does anyone know who this is? George Gershwin, very good. You're the first person who's ever gotten that. That's impressive. He happens to be my favorite composer, not for this reason, but nonetheless, this is George Gershwin, American composer, who died now more than 30 years ago, but back in 1937, at the age of 38, he died from a glioblastoma. This is Curtis Thomas. He's my late high school classmate, and he died from his grade 3 glioma in 2012 at the age of 24. This is clearly a devastatingly aggressive cancer, and it manages to evade treatment through a multitude of mechanisms. These are some examples of the genes that are altered in the glioma cells themselves. And as you can see, we are talking about some big hitters here, like the oncogene KRAS or the tumor suppressor gene P53. And these altered genes and more result in a highly malignant, locally aggressive, treatment-resistant cancer. Glioma's malignancy doesn't stop at the genetic level, though. I consider glioma to be a master puppeteer, exerting total control over the host immune system. In doing so, it prevents the host immune system from combating the tumor. Glioma controls both the local and the systemic immune environment. It's considered an immunologically cold tumor, meaning that it has a low degree of lymphocyte infiltration. The local tumor microenvironment is characterized by low levels of CD4 positive and CD8 positive T cells. And those that do get through are either exhausted or undergo anergy. Meanwhile, there is a markedly disproportionately high level of regulatory T cells, and then macrophages and microglia that are polarized towards the immunosuppressive M2 phenotype. There are also large degrees of infiltrating myeloid-derived suppressor cells. Glioma does not stop at the local immunosuppressive level. It also leads to systemic immunosuppression. Glioma causes lymphocyte sequestration in the bone marrow. And patients with glioblastoma experience lymphopenia that is worse than patients with AIDS. The circulating immune cell population is disproportionately immunosuppressive. There are marked increases in these regulatory T cells, and there are up to 12 times the level of myeloid-derived suppressor cells in circulation when compared to healthy individuals. Pictured here are some of the reasons that high-grade glioma is so resistant to treatment. And one of the biggest challenges in creating treatment is that glioma is a highly variable tumor. In an individual patient, no 2 biopsy sections look alike. And between patients, no 2 samples look alike. The heterogeneity makes it very difficult to create a treatment that will be consistently effective. So I think that some questions that are important to consider are these. What differences are there among patients pertaining to tumoral heterogeneity and immune evasion strategies that contribute to poor outcomes and may reveal possible novel treatment targets? And building on that question, is there a way that we can both control for and study this heterogeneity? Because doing so would allow us to identify subpopulations that may be candidates for one targeted therapy or another. And this is where the dog enters the equation. Canine high-grade oligodendroglioma is an aggressive brain tumor in dogs. Genetically, it is more similar to human glioblastoma than it is to human oligodendroglioma. In addition to sharing tumor genetics, the local immune landscape of canine high-grade oligodendroglioma mirrors that of human glioblastoma. Now, previous studies have characterized canine oligodendroglioma and have compared canine oligodendroglioma to human glioblastoma. But in those studies, all dog breeds have been lumped together. Is this the right approach? Are all dog breeds at equal risk for glioma? Are all dog gliomas the same? Are all dog gliomas equally good models for human glioblastoma? as I'm sure you can surmise from context clues, the answer to all these questions is no. We're going to pivot and talk about dog breeds, something that I find very enjoyable. And I presume, though, that this is something that most of you don't spend a lot of time considering, unless you are a veterinary medical professional or you're a dog breed enthusiast. The American Kennel Club is the largest registry of purebred dogs. It was founded in 1884 to maintain pedigree records to organize things so that dog sports were more regimented. The American Kennel Club currently recognizes over 155 dog breeds. Now, there are more than 155 dog breeds globally, but this only further drives the point that there are lots of versions of the domestic dog. The overall interbreed genetic variation in dogs is 27.5%, which is why a dog can look like a Chihuahua on one end of the size spectrum and a Great Dane on the other end. Or, pertaining to nose length, it can look like a pug on one end of the spectrum or a husky on the other end. Does anyone want to guess what the genetic variation is in people? I've got 6.1% and 1%, less than 5. You're all pretty close. It's 5.4%. Yeah. Selective breeding is responsible for making German Shepherds look like German Shepherds and Labrador Retrievers look like Labrador Retrievers. However, creating desirable breed-specific traits comes at a price. Within a given dog breed, only 10 to 14% of the population creates the next generation. And to maintain breed-specific or breed-associated traits, there's a high degree of inbreeding. Then, given that societal preferences for dog breeds wax and wane, genetic bottlenecks can occur where there are crashes in breed popularity. Then, if there's a sudden increase in breed demand that combines with that limited breeding stock and a narrow genetic pool, the result is a large, relatively homogeneous dog breed population and diseases that are along the ride as breed-associated or bred-in diseases. Here, I'm just showing you some examples of breed-associated diseases. Some breeds are notorious for certain cancer types. Like, for example, here, this is a flat-coated retriever. They are the poster child, among a couple other breeds, but the poster child for histiocytic sarcoma. Others are notorious for neurodegenerative diseases, like the Pembroke Welsh Corgi, which is the poster child for degenerative myelopathy. And this is the canine equivalent of amyotrophic lateral sclerosis or Lou Gehrig's disease. So you probably know where this is going. Are there dog breeds with a higher risk for glioma? And the answer is most definitely. This is a cladogram that shows all the dog breeds that are recognized by the AKC right now. And this group here in blue is the bulldog clade group. And this group includes dogs that are extremely popular right now. You probably see these on the street a lot. You might actually have one of these yourself. Up to 50% of gliomas occur in one of these bulldog clade breeds. The most popular bulldog clade breeds right now are boxers, Boston Terriers, French bulldogs, and English bulldogs. As in human glioblastoma, we have few effective treatment options for canine oligodendroglioma. Here, I'm reporting the survival data across presumed gliomas because we don't always have histopathological confirmation of the tumor type in dogs. As you can see over here with palliative care, which consists of steroids and anticonvulsants for dogs having seizures, the median survival time is 30 to 60 days. Given the diffuse nature of these tumors, surgery doesn't lead to any survival benefit over palliative care alone. The median survival time that's reported for dogs with gliomas treated surgically alone is 66 days. The median survival time with radiation therapy ranges from about 200 to 600 days, depending on the report that you read, the type of radiation therapy that's performed. For example, the median reported survival time following stereotactic radiation therapy is about 350 days, whereas it can approach 600 days for fractionated therapy. What we know is that these canine gliomas are aggressive and that we have few treatment options that are available. Those that are available are a one-size-fits-all. They're expensive and they're invasive, and there are many unexplored treatment targets. Dogs are also a strong large animal spontaneous model of glioblastoma. They have similar coevolution of their tumors in the tumor microenvironment. They have similar tumor heterogeneity. They have an intact blood-brain barrier that we have to battle with to get drugs passed. And especially important for today's discussion, they have an intact immune system. To help dogs with high-grade glioma and people with glioblastoma multiforme, the University of Minnesota Canine Brain Tumor Program investigates innovative treatments for pet dogs with spontaneous brain tumors. We focus primarily on gliomas, and today I'll be discussing dogs that were enrolled in our immunotherapy clinical trials. So the dogs that I'll be discussing today were enrolled in a clinical trial that consisted of surgical resection to remove as much of the tumor as possible. This also allowed us to obtain histopathologic diagnoses and to create autologous tumor lysate vaccines. They all underwent immediate postoperative MRIs to evaluate the extent of tumor resection. From the resected tumor tissue, we created these autologous tumor lysate vaccines that we then administered subcutaneously. The premise of this therapy is that the autologous tumor lysates contain tumor-associated antigens and neoantigens. Dendritic cells in the dermis identify these neoantigens, which leads to dendritic cell activation and homing to a lymph node, where the dendritic cell can then interact with naive CD4 positive and CD8 positive T cells. Those T cells are then activated to participate in an immune response against the residual tumor cells. Now, I think we do a good job of removing these tumors, but I guarantee you, whenever we do surgery, we leave tumor behind. There's just no way to get full, complete margins when you're dealing with a brain tumor in dogs. The problem was that among the many antigens that were being delivered to these cells, immunosuppressive immune checkpoint molecules were being administered. CD200 is an immune checkpoint ligand that glioma secretes in very high concentrations, and it is highly immunosuppressive. When CD200 binds to its receptor on immune cells, it inhibits downstream pathways, which thereby prevents immune cell activity. And essentially what that meant was that administering these autologous tumor lysates meant that we were delivering CD200 to the very immune cells that the autologous tumor lysates were aimed to activate. Excuse me, I'm getting over a cold. CD200 ARL is a molecule that Mike Olin and colleagues created, which outcompetes with CD200. And it enables the dendritic cells to respond to the vaccines. And sure enough, when CD200 ARL was added to the autologous tumor lysate therapy, it doubled the survival time in dogs from about 6 months to about 12 months. However, unfortunately, this wasn't the case for every dog. It turns out that the first 3 dogs that were enrolled in this study did very poorly. And when we looked back, all of them happened to be one specific breed, the French Bulldog. When we looked back, we realized that in retrospect, French Bulldogs in previous immunotherapy trials that we'd conducted had also done poorly. And this wasn't true for other Bulldog clade members who had done very well with the immunotherapy treatments. This Kaplan-Meier curve here is from a retrospective study that we performed where we examined survival time by breed in dogs that were enrolled in various immunotherapy clinical trials for their gliomas. And as you can see, there was a significant difference in survival time. French Bulldogs had a median survival time of 132 days, whereas Boxers and Boston Terriers had a median survival time of 221 days. This meant that we excluded French Bulldogs from further enrollment in the trial because it wasn't helping them. Why did we have 3 French Bulldogs in a row? Well, it wasn't just coincidence. French Bulldogs are currently the most popular dog breed in the United States. In the early 2000s, it was a relatively obscure breed, but then their popularity completely exploded, honestly, seemingly overnight. I remember I was a resident when things started getting concerning involving how frequently we were seeing these dogs. And I remember telling my faculty at the time, we're seeing a lot of these French Bulldogs, and he's like, nah, I don't think so. I was like, no, I'm worried. I think these are becoming more popular. The reason I was worried is because these dogs have a lot of health problems. We all as trainees think we know everything. In this case, I was right. and they became super popular. And a couple years after this all happened, he called me one day and he was like, yeah, I'm sorry, you were right about this. You were right to be concerned. And it's really fascinating because up for the past 30 years, leading up to 2022, the Labrador Retriever was the most popular dog breed in the United States for 30 years in a row. And then the French Bulldog came along and dethroned it. To meet the demand and to create all sorts of types of French Bulldogs, there's been a lot of inbreeding that's occurred and continues to occur in this breed. The coefficient of inbreeding in French Bulldogs is 28 to 30%. For reference, a mating between full siblings yields a coefficient of inbreeding of 25%. My point here is that the French Bulldog is an especially tightly genetically linked breed, and there's not a lot of diversity in the genetic makeup among French Bulldogs. We know that both human and canine high-grade glioma are highly heterogeneous. Maybe what we can do is we can take some of that heterogeneity away, take it out of the equation, by considering dog breed. Doing so would help us answer the questions that I posed earlier. Does canine high-grade glioma have breed-specific features? Do some dog breeds have more aggressive tumors than others? And are some dog breeds better models for glioblastoma? We also know that glioma is highly immunosuppressive. Although there has been some work done to characterize the immune response in dogs, again here, breed has not been considered. And there hasn't been a lot of work done on investigating the systemic immune response in dogs with glioma. To address these knowledge gaps, our goal is to establish breed-specific canine models of glioma. Because the French Bulldogs did so poorly in the clinical trial, and because they are so popular, we chose them as our first breed to characterize them. Oh, thank you. Ultimately, we want to do the same with other dog breeds, as I'm showing here, so that we can create ultra-specific models for human glioblastoma. Okay. This leads us into part 2, which is the characterization of the canine glioma transcriptome. My second fun dog fact for you is that just like human fingerprints, dog nose prints are unique. So what did we do here to start evaluating the tumor transcriptomes of French Bulldog oligodendrogliomas? We hypothesized that compared to other bulldog clade breeds, French Bulldogs have clinically important transcriptomic differences in their tumors and the tumor microenvironment, as well as in the systemic immune response to high-grade glioma. This could explain the survival disparity in the French Bulldogs in our studies. We chose to use Boxers and Boston Terriers as our comparison group throughout our studies for several reasons. They're members of the same phylogenetic clade as French Bulldogs. They have similarly high breed-associated risk of high-grade oligodendroglioma. They're common breeds in our clinical trials, and they did not seem to have the same poor survival outcome as French Bulldogs. To examine our hypothesis, we used formalin-fixed paraffin-embedded tumor tissue from French Bulldogs and Boxers and Boston Terriers. We took pre-treatment samples that were obtained at the time of surgical resection that was done prior to immunotherapy. We also obtained post-mortem samples that were obtained from the dogs after they've been treated with the autologous tumor lysates and CD200. And then we performed 2 different analyses. First, to determine if French Bulldogs and Boxer and Boston Terrier oligodendrogliomas are inherently different, we investigated differential gene expression using the pre-treatment samples. Second, to determine if French Bulldog and Boxer and Boston Terrier tumors changed differently in association with immunotherapy, we investigated the post-treatment samples compared to the pre-treatment samples by breed, and then we examined the differences between the breeds. The University of Minnesota Genomics Center performed bulk RNA sequencing for us using the CanFam6 as the reference genome. And then we performed differential gene expression using the DSeq2 package for R. We designated that an absolute log2 fold change greater than 1, which is the same as a fold change greater than 2, was our cutoff for biologically relevant differentially expressed genes. We also performed gene set enrichment analysis to determine if our genes were enriched or depleted in the sample populations. So here's what we found. Again, these are pre-treatment transcriptomes, so these are dogs that have not seen any immunotherapy yet. French Bulldogs versus Boxers and Boston Terriers. There were 31 protein coding genes that were differentially expressed between French Bulldogs and Boxers and Boston Terriers. 14 of them were upregulated in French Bulldogs and 17 were downregulated. Pertaining to gene expression, the samples grouped by dog breed, with French Bulldog samples all clustering together and Boxers and Boston Terriers all clustering together. These are some of the notable differentially expressed genes that were observed in this analysis. I'm not going to discuss all of them, but I do want to discuss the relevance of the upregulation of FGF4 and DLA88. Fibroblast growth factor 4 is a growth factor. It binds to receptors that are associated with many pathways, including the PI3K, AKT, mTOR pathway, and the MAP kinase pathway. Upregulation of FGF4 is associated with tumor growth promotion through increased activity of these pathways. This I find super fascinating. In dogs, the FGF4 gene has been studied most extensively in the context of chondrodystrophy. Chondrodystrophy is a condition in which animals have altered skeletal development. They have short noses and they have, not short noses, they have short legs and long backs, angular limb deformities. Think of breeds like Dachshunds, American Cocker Spaniels, and Basset Hounds. French Bulldogs also have a retrogene version of FGF4. And they have an extremely high allele frequency of the retrogene. 90% of dogs carry, 90% of French Bulldogs carry one of these retrogenes. In these dog breeds, the FGF4 retrogene is not regulated like the normal native version of the gene. The dysregulation is what leads to the skeletal growth abnormalities that are observed in these dogs, which make them have these short legs and long backs. Boston Terriers do not have the retrogene, and we can assume that neither do Boxers. FGF4 overexpression in the French Bulldogs in my study could just be a breed-associated variant. But one thing that we considered instead was this. The FGF4 retrogene doesn't cause French Bulldogs to experience high-grade glioma, because if it did, then the glioma would also be more common in other dog breeds with the FGF4 retrogene, which it's not. We don't see gliomas commonly in Dachshunds, in American Cocker Spaniels, or in Basset Hounds. But in the French Bulldog, is increased FGF4 expression something that is fueling these pro-tumorigenic pathways that drive a more aggressive high-grade glioma version than Boxers and Boston Terriers? I'll move on to another noteworthy upregulated gene, which was DLA88. This is dog leukocyte antigen 88. It's a major histocompatibility complex class 1 molecule. And as such, it's expressed on many cell types, including macrophages, fibroblasts, endothelial cells, to name a few. And its job is to present antigens to cytotoxic CD8 positive T cells. Does this perhaps mean that there are an increased numbers of infiltrating immune cells in the French Bulldog tumor microenvironment? Alternatively, glioma cells can upregulate their MHC class 1 molecules to evade natural killer cell detection. Does this mean that perhaps the French Bulldog high-grade oligodendroglioma employs this defense strategy more than Boxer and Boston Terrier samples when it pertains to oligodendroglioma? Here, I'd like to perform some single-cell RNA sequencing to determine which cells are responsible for this upregulated gene in the French Bulldogs. MHC class 1 molecules have an extremely high degree of polymorphism. In people, an individual's HLA polymorphism impacts their responsiveness to cancer therapy. An example would be HLA A*03. This is associated with reduced survival in people treated with immune checkpoint therapy. And just as in people, there's a high degree of DLA polymorphism in dogs. There have been 139 alleles reported to date. And the DLA88 profile has been characterized in Boxers thus far, but not in Boston Terriers or French Bulldogs. Purebred Boxers all have the same dominant allele as a result of genetic bottlenecking. It's equally possible and highly likely that French Bulldogs have their own single dominant allele, given how much genetic bottlenecking there's been in French Bulldogs. Since treatment responsiveness is linked to polymorphisms in people, the same could be true for dogs. Perhaps French Bulldog high-grade glioma has a specific polymorphism that negatively correlates with prognosis. Beyond these specific important differentially expressed genes, there were important pathways that were upregulated in French Bulldog tumors compared to Boxer and Boston Terrier tumors. This is a dot plot that shows that there were 15 pathways that were upregulated in French Bulldogs compared to Boxers and Boston Terriers. The pathways can be organized into 3 broad categories. We have cell cycle progression pathways. It's going to be hard to see here, but things like E2F targets, G2 to M checkpoint, and the mitotic spindle are up there. We had upregulation of oncogenes like KRAS signaling, the mTOR signaling pathway, and MYC targets. And then there were a lot of pathways involved in immune regulation, like the IL-2 STAT5 signaling pathway, TNF alpha signaling via NF-kappa B, and allograft rejection, just to name a few. This is a gene concept network plot, and it visualizes overrepresentation analysis. And in this type of an analysis, significantly differentially expressed genes are mapped to pathways. What it shows here is that among the identified significant genes, many of them were associated with the PI3K AKT signaling pathway and also with cell cycle. Investigation of these upregulated pathways reveals that many of them are similarly upregulated in human glioblastoma and are negatively associated with prognosis in people. E2F, for example, is a transcription factor that is both an upstream signal and a downstream target of P53, and it guides the cell toward proliferation. E2F dysregulation is a strong hallmark of tumor proliferation in glioblastoma. The G2M cell cycle checkpoint marks the final opportunity for cells to either proceed through mitosis, undergo arrest, or go through apoptosis, depending on the extent of DNA damage. And as with E2F, upregulation of this pathway is a hallmark of glioblastoma progression. This shows the RAS signaling pathway and the PI3K AKT mTOR pathway. Collectively, these intersecting pathways promote cell growth, division, and differentiation. And KRAS is the most commonly mutated form of RAS in glioma. mTOR is frequently dysregulated in human glioblastoma and correlates negatively with prognosis. Interestingly, mTOR has not been shown to be elevated in canine glioma cell lines, in non-French Bulldog canine glioma cell lines, at least. So its upregulation in French Bulldogs could be especially meaningful. I want you to remember this upregulation of mTORC1 for later, because it's going to show up again and again. And then MYC is amplified in the majority of our studied canine glioma samples, and in human glioma, expression correlates with glioma grade. The largest category of enriched pathways was centered on immune regulation. What this suggests is that the tumor immune interaction of French Bulldogs is prognostically different from that in other breeds. And many of these pathways have known tumor driving roles in glioma. For example, TNF alpha induces endothelial cell activation to promote tumor survival. So from these pre-treatment immunotherapy naive samples, our conclusions were that there are important differences in gene expression between the breeds, and that French Bulldogs have upregulation of numerous pathways that are negatively correlated with prognosis in human glioblastoma. And many of these pathways involve the immune system. I'm going to pivot now to talk about our post-treatment versus pre-treatment analysis. We wanted to do this because we wanted to determine how the tumors changed at the transcriptome level following immunotherapy, as we know that these tumors are highly dynamic and evolve in response to treatment. For this investigation, we performed 2 paired differential gene expression analyses using DE-seq2. We determined the differentially expressed genes of French Bulldogs post-treatment versus pre-treatment, and then we did the same for Boxers and Boston Terriers post-treatment versus pre-treatment. Now, these were paired analyses, meaning that each dog was compared to itself post-treatment versus pre-treatment. After we performed those 2 analyses, we combined the data sets. We performed a Descartes index calculation to determine the percentage of differential gene expression changes that were shared or were specific to one breed versus the other. We performed a variety of visualization techniques to identify patterns in differential gene expression. And then we determined the differences in the log 2 fold changes between French Bulldogs and Boxers and Boston Terriers. And here's what we found. In the French Bulldog differential gene expression analysis, there were 488 genes that were upregulated and 269 that were downregulated post-treatment compared to pre-treatment. In the Boxer and Boston Terrier analysis, there were 1,749 upregulated and 1,924 downregulated genes post-treatment compared to pre-treatment. The top 2 heat maps here are the outputs for these 2 analyses. This one here is the expression heat map for the French Bulldogs, and this one is the expression heat map for the Boxers and Boston Terriers. Again, looking at post-treatment versus pre-treatment genes. And as you can see, the patterns are different. The bottom 2 heat maps here show the expression of one breed's differentially expressed genes in the other breed group. This one here is the expression pattern of French Bulldog genes in Boxers and Boston Terriers, and this one is the expression pattern of Boxer and Boston Terrier differentially expressed genes in French Bulldogs. Essentially here, I'm just trying to further drive the point that the tumors look different from each other by breed, and that they change differently in response to or in association with treatment. The Jacquard index is a calculation to determine the proportion of shared differentially expressed genes. And a Jacquard index of 0.159 means that there was only a 15.9% overlap in the significant differentially expressed genes between French Bulldogs and Boxers and Boston Terriers. These are 4 bar plots that show the outputs of the differential gene expression analyses. This one here shows the top 25 significantly upregulated genes in post-treatment French Bulldogs. This one shows the top 25 significantly upregulated genes post-treatment in Boxers and Boston Terriers. Oh wait, let me make sure I'm getting that right. Yeah, sorry, it's kind of tiny up there. This one shows the top 25 significantly upregulated genes in Boxers and Boston Terriers, and this one shows the top 25 downregulated ones in Boxers and Boston Terriers. In each of them, French Bulldogs are in pink and Boxers and Boston Terriers are in blue. What we observed overall was that there's little overlap in the top 25 differentially expressed genes of French Bulldogs versus Boxers and Boston Terriers, both for upregulated and downregulated genes. And there were some important patterns that emerged. So we'll start by looking at the top 25 upregulated genes in French Bulldogs. These genes have a higher magnitude of log 2 fold change in the French Bulldogs than they do in Boxers and Boston Terriers. An opposite pattern emerges when we looked at the downregulated differentially expressed genes. Among the top 25 downregulated genes in French Bulldogs, the magnitude of downregulation was actually larger in the Boxers and Boston Terriers than it was in the French Bulldogs for most of the genes on this plot. Similarly, the magnitude of downregulation in the top 25 downregulated differentially expressed genes in Boxers and Boston Terriers was much higher than in French Bulldogs. Is it possible that the larger magnitudes of French Bulldog upregulated genes means that their tumors are escaping immunotherapy? And is it also possible that the larger magnitudes of Boxer and Boston Terrier downregulated genes means that their tumors are responding to immunotherapy, more so than French Bulldogs? To further identify genes that define the French Bulldog specific tumors, we determined the top 25 upregulated genes that were differentially expressed in French Bulldogs, but were not differentially expressed in Boxers and Boston Terriers. And those 25 genes are shown here. Interestingly, there were 4 upregulated genes in French Bulldogs post-treatment that were actually downregulated in post-treatment Boxers and Boston Terriers. BAG3 is one of those. That's just an example to give. It's an anti-apoptotic protein that's upregulated in human glioblastoma. So potentially, there's a mechanism here by which immunotherapy in Boxers and Boston Terriers is associated with downregulation of BAG3, but fails to downregulate the gene in French Bulldogs. There were other important differentially expressed genes to note here as well. For example, GPRC5A is upregulated in human glioma, and 6,4 promotes angiogenesis in glioma. These are the dot plots for the gene set enrichment analyses, looking at post-treatment versus pre-treatment. Here are the French Bulldog genes, and here are the Boxer and Boston Terrier genes. In French Bulldogs, there were 7 upregulated pathways and 5 downregulated pathways. In Boxers and Boston Terriers, there were 6 upregulated pathways and 8 downregulated pathways. Now, there was some overlap in the important enriched pathways between the two breed groupings, including MYC targets, the G2M checkpoint, E2F targets, and allograft rejection. But there were several important pathways that were only activated or suppressed in one group. And the one that I'm going to harp on again is mTORC1. mTORC1 signaling was suppressed in Boxers and Boston Terriers, but was not suppressed in French Bulldogs. And I'll just remind you from the pre-treatment samples, mTORC1 signaling in French Bulldogs was enriched. What we conclude from this evaluation is that compared to Boxer and Boston Terrier high-grade oligodendroglioma, French Bulldog high-grade oligodendroglioma has a distinct transcriptomic profile and experiences transcriptomic changes differently post-immunotherapy. The changes that we found were involving genes that are known to be dysregulated in glioblastoma, and there was a large proportion of alternate immune pathways. And then further, something very interesting emerged when we compared the results between the pre-treatment analyses and the investigation of our post-treatment transcriptomic changes pertaining to mTORC1 signaling. This is what we observed. As pertaining to mTORC1 was upregulated in our pre-treatment French Bulldogs compared to our pre-treatment Boxer and Boston Terriers. And it was downregulated in our post-treatment Boxer and Boston Terrier samples compared to the pre-treatment Boxer and Boston Terrier samples. Does upregulation of mTORC1 in French Bulldogs compared to Boxers and Boston Terriers contribute to a more aggressive form of high-grade glioma in French Bulldogs? And does mTORC1 downregulation following immunotherapy in Boxers and Boston Terriers correlate with their superior response to immunotherapy? So now we're going to pivot and talk about the systemic immune response in the presence of glioma. To lead us off, I'll finalize this section with fun dog fact number 3, which is my favorite. Petting a dog for 15 minutes can lower your blood pressure by 10%. So go pet a dog. So in addition to characterizing the transcriptomes of the tumors, we also sought to compare the systemic immune profiles of French Bulldogs versus other breeds. The rationale here is that local immunosuppression has been observed in both human and canine tumors. Systemic immunosuppression has been observed in human glioblastoma, but there's yet to date minimal characterization of canine systemic immune response to high-grade oligodendroglioma. We hypothesized that high-grade oligodendroglioma-bearing French Bulldogs experience systemic immunosuppression compared to healthy French Bulldogs. I'll put that in quotes. There's not really such a thing as a healthy French Bulldog. And they also experience a different systemic immune response than other dog breeds. We obtained whole blood samples that we stored in Tempest tubes prior to our analysis. We then performed RNA extraction, and then we used NanoString using their canine immuno-oncology panel. The canine immuno-oncology panel contains 800 genes across 47 annotated pathways that pertain to immuno-oncologic processes. And the panel also includes 20 housekeeping genes as internal reference. We then performed differential gene expression analyses in R. Now, so far to date, I only have relevant data that compare tumor-bearing French Bulldogs to healthy French Bulldogs. I need to obtain more samples from other dog breeds to compare the systemic immune profiles of French Bulldogs to other dog breeds and to other healthy breeds. But for now, I just want to share these data because I found it interesting. And the reason I think it's interesting is that really right off the bat, we just don't know a lot about how the canine systemic immune response responds to oligodendroglioma. And we definitely don't know how breeds have differences. So again, we started with French Bulldogs because they were our concerning breed population. Between tumor-bearing and healthy French Bulldogs, we found 54 differentially expressed genes. There were 8 upregulated genes, and there were 46 downregulated genes. I'm just showing the top 25 downregulated genes here. And the expression heat map showed that mostly dogs with tumors clustered together and that healthy dogs clustered together. In French Bulldogs with tumors compared to those without tumors, there were over 5 times more downregulated genes than upregulated genes. We categorized the genes by function, And what I'm showing you here in pertaining to the downregulated genes is that they're involved in T cell and B cell function, cell migration, broad immune functions, and cellular repair and apoptosis. Conversely, 6 of the upregulated genes were involved in either general immune function or the innate immune system, and 2 of them were pertaining to non-immunologic pathways. Overall, what we conclude is that the higher degree of downregulated genes in French bulldogs with tumors suggests that at least in French bulldogs, high-grade glioma is associated with immunosuppression at the RNA transcript level. And the gene set enrichment analysis that we performed further heightened this suspicion. There were 6 upregulated pathways, and only 2 of those were immune pathways. And there were 9 downregulated pathways, all of which were immune pathways. We suspect that there are several mechanisms at play that lead to these downregulated pathways. Immunosuppressive cellular factors like TGF-beta, IL-6, and VEGF interfere with dendritic cell maturation. This then goes on to reduce antigen presentation, which interferes with lymphocyte activation and gamma delta T cell activation, which were both identified pathways in our analysis. Immune checkpoint proteins lead to decreased T cell and B cell signaling, also downregulated for us. Increased circulation of myeloid-derived suppressor cells could lead to reduced B cell and T cell proliferation and receptor signaling, and gamma delta T cell activation, all of which were downregulated. We didn't quantify myeloid-derived suppressor cells or other cell types, but recent studies have confirmed that, like in people, dogs with cancer have elevated levels of circulating MDSCs. The PI3K-AKT-mTOR pathway signaling was upregulated in French bulldog tumor samples pre-treatment compared to boxers and Boston Terriers. And as I've talked about, mTORC1 signaling was upregulated in French bulldog tumors compared to boxer and Boston Terrier samples. That mTORC1 pathway was downregulated post-immunotherapy in boxer and Boston Terrier samples. The PI3K-AKT-mTOR pathway not only upregulates tumor-promoting expression, but also leads to the downregulation of various chemokines. By this point, hopefully you realize I'm very interested in this pathway, especially mTORC1, as a key differentiator of French bulldog high-grade gliomas compared to other breeds. This is why it excites me. The chemokine CCR7 was one of the downregulated genes in the circulating immune cells of this analysis here. CCR7 is downregulated by the PI3K-mTOR pathway. Downregulation of CCR7 in people contributes to bone marrow sequestration of T cells. Remember this? There you go. Is that potentially contributing to bone marrow sequestration of T cells in French bulldogs? So overall, what we concluded here was that compared to our healthy French bulldogs with tumors had downregulated immune profiles, which overall suggests immunosuppression. And again, I'm very interested in following mTORC1. With that, I'll summarize what our overall conclusions were. We conclude that French bulldogs treated with immunotherapy for their tumors have a uniquely poor outcome compared to other breeds and to other treatments. French bulldog tumor transcriptomes are characterized by more aggressive cancer hallmarks and a more immunosuppressed tumor microenvironment compared to boxers and Boston Terriers. And their tumor transcriptomes change differently following immunotherapy, with many enriched immune pathways. Finally, French bulldogs with high-grade glioma showed transcriptomic evidence of systemic immunosuppression compared to French bulldogs without tumors. There are many exciting directions that I want to take this research going forward. And here are some of the future directions I've considered so far. First, we're going to focus on validating our findings to enable us to identify potential therapeutic targets. Right now, we're especially interested in FGF4, DLA88, and most importantly, mTORC1 signaling. Validation will consist of Western blots and functional analyses, like functional assays. Alternatively or additionally, we're considering performing spatial transcriptomics or single-cell RNA sequencing to correlate gene expression with cell types. We also want to combine the transcriptomic evaluation of the peripheral immune system with other descriptive techniques. For example, looking at complete blood counts in these dogs, maybe performing flow cytometry as well. To further refine the descriptions of breed-specific canine high-grade glioma, we're planning on obtaining blood samples from boxers and Boston Terriers with tumors so that we can compare the peripheral immune response by breed. We also want to examine how genomic alterations can lead to breed-specific versions of high-grade glioma. Now, the ultimate goal of this research is to be able to compare our findings with human glioblastoma samples so that we can validate these breed-specific canine models for subsets of glioblastoma patients and develop breed-specific translational therapies. I want to thank you all for attending my talk today. Thanks for your attention, and I'm happy to take any questions.
Thank you, Susan.
I have microphones for anyone with a question.
Carol.
Hi, thank you. So many of us steroid receptor people were at a recent Gordon conference, and one of the speakers, Donald McDonald, showed that in these models that also have a lot of connection to MAP kinase and mTOR signaling, say lung cancer, melanoma, that estrogen, the hormone estrogen, dramatically inhibited the NK cells and one of the macrophage populations, and that he could treat preclinical models with tamoxifen and see really good activity in these cancers that aren't really. So he knocked out the ER gene in the cancers and then showed that it's really an extrinsic function of ER in NK cells and macrophages that was suppressing the immune system. So I'm just wondering if aromatase showed up in your list, or I don't know that it was really upregulated at all in these tumors. He didn't really show that. I don't know if you remember, Scott, but it was really cool. So it suggests that maybe you can use endocrine therapies in other cancers.
Yeah, thank you for that. That's really interesting. Aromatase did not show up in my analyses, but I will consider it. And this is a question that I've encountered before pertaining to sex hormones. And all of the dogs in this study were altered, meaning they were spayed or neutered. And I don't know how much that would impact, they're certainly going to have some degree of circulating sex hormones, but how much they have is dramatically impacted by being altered. Do you recall in those, I would presume those models were mice, rodents of some sort? Okay, yeah. It's certainly worth looking into. I haven't done that, and I don't remember aromatase coming up, but I will write some notes about that. Thank you.
You had an ER pathway, one of the slides.
I had an ER pathway.
Well,
I have E2F.
The androgen response pathway was uniquely downregulated in the boxers and terriers group.
I'm sorry, you're right. I do have that. Yes.
So
yes,
I think that's something to look into, definitely based on that question that Carol had asked. I had a question about the sampling itself. So when exactly and how exactly were these post-treatment samples collected? And is it similar to or different from the pre-treatment samples? And what was the tumor cellularity? All those sorts of technical factors that can impact the quality of data like this.
Yeah, that's a great question. So the post-treatment samples were taken from, so we harvest whole brain after the animals are euthanized or they die. And then we section the brains, and they're all, they're all formalin fixed blocks, I should say. And so what I did was I went through all of the blocks and I selected areas that had tumor on them. And so as much as we could, we compared tumor to tumor. Now, you're getting to a good point. There is certainly much more opportunity for time-associated artifactual changes because those brains could sit for a much longer, potentially, period of time before they were sectioned. That's ultimately why I did the paired analyses as well, was to be able to compare dog to dog to help with some of that. But ultimately, what we concluded was that just looking at the RNA integrity numbers, they were quite similar between our pre and post-treatment samples.
And then I just have one more question. with dogs, is there some sort of a bias in terms of the dogs that get enrolled on the trial, as opposed to dogs who would just be euthanized by their owners because of this diagnosis instead of put on therapy? So are you kind of selecting for a population of dogs that might be different than just the standard dog that would be getting a glioma?
That is really interesting. And maybe I'll start by asking a question. Are you thinking potentially that dogs that are owned, how am I put this? People who have specific dog breeds might be inclined to pursue treatment of one form versus another?
Yeah, or an owner who is willing to go to the ends of the earth to keep their dog alive, may have fed them more expensive or better food throughout their life, and provided different care. I'm just wondering if there's any sort of a bias just based upon the sorts of dogs that you would see in these sorts of trials compared to, say, a human.
Yeah, that's very interesting. And I think it's probably a wonderfully complicated conundrum. And because if I just think about our patient population, we get people from all over the country. So geography doesn't seem to be a huge factor. We also get dogs. So most of the people who enroll their dogs in our clinical trials do so, frankly, because they can't afford definitive treatment. But they are a highly motivated group of people. I don't know if it necessarily correlates to how they've taken care of their dog in the past, because sometimes when people get this type of diagnosis, they might have, loved their dog a lot, but now they love their dog a lot. And so it can the human behavior part of it, I think, would be really challenging to suss out there. Yeah.
So I don't know how much the tumor type influences the question, but one of our residents, Dr. Luthke, did a study trying to understand socioeconomic factors that might impact lymphoma in the population at the veterinary medical center. And in a nutshell, his conclusion was that owners of dogs with lymphoma that were treated at the veterinary medical center were all in the top quintile of income. And so, you can reach a lot of conclusions from that. I think that the average cost to treat a dog with lymphoma with palliative care is pennies, because you put them on prednisone, and to go full chemo is about 10 grand. Whereas the average cost to treat a full treatment on a dog with brain cancer is about 20 grand.
20,
Yeah. So there's a big gap there. And I think that the motivation for owners to be able to enroll in a clinical trial that pays for everything might actually reduce some of the bias. But that was a very surprising result to us, that we think we attract people from all walks of life, and maybe not. Sorry.
Thanks.
Just a quick question. That was a great talk. Thank you.
Thank you.
Very intriguing for those of us who may own boxers. So I was actually really intrigued by your FGF4 findings. I have a longstanding interest in FGFR signaling in cancer. And there's some pretty good FGFR inhibitors out there, some of which are in clinical trials in humans. Is that a direction you might be interested in?
I would love to go in that direction. So thank you for just a really great plug for the canine brain tumor program. So as Dr. Modiano mentioned, I just took over as a director, and I've gotten to take over at a really exciting time in the brain tumor program, because we are at a point where we've concluded a lot of our trials, and we're now in the early phases of we have a new one right now that we're treating dogs in. But ultimately, my vision for the canine brain tumor program is to be a platform for researchers like yourself to come to the canine brain tumor program and say, I've got this product that, we've gotten it to a point where we want to put it in animals, and we want to put it in people. Before we put it in people, we want to test it in dogs, because we know there's value for the dogs, there's value for people. So if that's something you want to do, I am all for it.
Great.
Yeah.
And just one comment. So there's some data coming out of the breast cancer field, at least, and some other tumor fields, that FGF receptor signaling is actually quite immunosuppressive as well. So that might fit into your immunosuppression.
Amazing.
Category.
Thank you.
All right, I think we've got a question from Dougie.
Yeah, hi, thanks. That was a really great talk, and appreciate Scott's question about the social determinants of health for dogs. That was kind of an interesting sidelight. But, I was going to follow up on Kaylee's comment. So in humans, achondroplasia is caused by an activating mutation in FGFR3. So I guess dogs do it the same way, but with a different mechanism, but they're probably activating FGFR signaling. And to me, and I'm a kind of a tumor intrinsic growth factor person, it seems like your mTOR activity was related to that. So during your talk, I think you were probably not emphasizing the tumor intrinsic signaling pathways activated by the gliomas themselves. And I bring it up because there are tons of rapalogs and PIK3CA and AKT inhibitors that are floating around there in human clinical trial. I do not know the cross-reactivity with dogs, but it would be easy enough, I think, as Kaylee mentioned, to set up an FGFR inhibitor with a PIK3CA mTOR inhibitor and see if that has activity. So anyway, that was a long prelude. You seem to be discounting a little bit the tumor intrinsic properties of the signaling pathways. I just wanted to get your thoughts on that.
No, I think they're great directions to pursue, and I just need to delve into it a little bit more at this point. So I really appreciate that comment because it's very motivating to pursue this further. Thank you.
All right, great. We are out of time. So let's say thank you one more time to Dr. Arnold.
Thank you.